Today we are covering a paper that covers a phenomenon or discusses a phenomenon that we discussed in a previous paper, silent synapses. If you will recall, silent synapses are synapses that do not contain any AMPA receptors but only NMDA channels, and therefore cannot be opened by glutamate release alone, but also require some depolarization to remove the magnesium block, and a silent synapse will mature into a functional synapse when AMPA receptors insert. So the context in which we're discussing silent synapses is in the context of ocular dominance, plasticity, and critical periods in the visual cortex. Those are fairly simple to summarize, but let's just say that in the binocular region of the visual cortex, where neurons receive input from both eyes, during the course of the maturation of the early visual system, what starts out as a very balanced binocular circuit ends up slowly maturing into a circuit where the contralateral eye dominates the visual field. Now, there's still ipsilateral input, but the contralateral eye is the eye that dominates the binocular region of any particular cell. And interestingly, during the critical period of visual cortex development, if you monocularly cover one eye, then the maturation of this contralateral bias can be disrupted. And you can essentially reverse this bias toward the contralateral eye back to a balanced neuron that receives or responds equally to both eyes, contralateral and ipsilateral. But interestingly enough, monocular deprivation will only cause this reversal during the critical period of the visual system. So if you do it before or after the critical period, then you cannot reverse this ocular dominance plasticity, and instead the contralateral eye will remain the dominant eye. So this paper, I think, is largely based on the observation that in PSD95 knockout mice, ocular dominance plasticity windows don't really exist, and in fact, critical periods in the visual system don't really happen there. Now, interestingly, it's not that these animals have a misfunctioning or a, let's say, non-functioning visual system. That's not the case at all. They have a pretty normal visual system, but ocular dominance plasticity and critical period of the visual cortex never really closes. And in fact, ocular dominance plasticity remains open into the adult life of the PSD95 knockout mouse. So that observation already cued these authors into thinking, well, what happens when this critical period closes? And so they've already known that the critical period closes in the same time period that these silent synapses are maturing. They already knew that. But this paper provides a couple unique methodological ways of testing what precise component of the pre and post synapse might be the critical component to determining whether these immature synapses, these silent synapses, mature into mature synapses and then remain that way. Because that's the trick, right? They have to remain that way, because once this critical period is closed, you cannot monocularly deprive the animal and rewire this change again. It just doesn't work. So not only is there a maturation of silent synapses that coincides with the closing of the period, the critical period, but once the synapses are mature, people have not found a very easy way to make this ocular dominance plasticity come back. It seems to, the window seems to close and then you can't do it anymore. But PSD95 knockout mice, mice without this protein in the postsynaptic density, which is thought to be a scaffold protein, they do not show this progressive maturation of silent synapses, and their critical period window doesn't close. So you can see where this is going to go. So in the first figure, they just show you the observation I just explained, that PSD95 knockout mice do not have a typical critical period window for visual cortex ocular dominance plasticity, but instead this window remains open into adulthood. And that figure is going to take you a while to digest, but you can do it. So now they're going to check this out to see what kind of observations they can make in the network of the PSD95 knockout mouse, to see if they can find a molecular or synaptic basis for this lack of critical period window closing. And what they find is that the ratio of silent synapses to mature synapses in the PSD95 knockout mouse doesn't really change over the course of the maturation process. And that's very, very, very different than what they see in the wild type animals, where maturation of these silent synapses is very, very measurable and quantifiable. The way they do it is that they do a stimulation in layer 4 and then they record excitatory postsynaptic potentials. It's not really clear to me exactly how this experiment works. I don't know if they move the stimulation electrode around in layer 4 or if there's some other way of generating the different responses that they then say they're, counting silent synapses and mature synapses. It looks to me almost as if it's just the failure rate that they quantify. Thank you. If the failure rate decreases, then the number of mature synapses is increasing. That's what it looks like, but it's not really clear to me how they do it. So it might be a good question to ask the author or the lecturer in this case. So figure 3 is like another kind of control figure. What they do with figure 3 is they look at various synaptic proteins to see if synaptic proteins in the wild type versus the knockout mouse are coming up at roughly the same time. And for the most part, they do see that, with the exception of AMPA receptors. The AMPA receptors are clearly not coming up at the same time and are not present, at least in the adult, that they are in the wild type. That's again just a molecular biology figure, but nonetheless, it's again trying to check what changes are responsible for bringing out the maturation of the synapses. What is this human doing? He doesn't know. no turn signal, and you were in the right lane. And you were in the right lane, and you went in front of me, even though you were supposed to go right. What are you doing? Are you going to ride your brakes all the way down the hill? So I have to ride my brakes all the way down the hill. No, thank you. I'm definitely not going to wait for somebody who runs a red light and goes the wrong direction out of the lane that she's in. Come on, go! Come on! What are you waiting for? Frickin' A! You make me stop and then you're not gonna go? It's crazy, these drivers, man. I'm telling you, it's crazy. So figure 4 is a really cool one because what they do is check the AMPA and MDA ratios again. But now they're going to look at AMPA and MDA ratios and show that the AMPA and MDA ratio changes specifically for pyramidal cells and not for fast spiking cells. So because it doesn't change for fast spiking cells, they can make the argument that indeed it is specifically the excitatory synapses on pyramidal cells that are changing. And then what they do is they also do a ratio of, god dang idiots. They do a ratio of AMPA to GABA to get a kind of an idea of whether GABA changes over time. And they don't see that. God, that's a junky car. That was a Porsche that sounded like that but didn't move. So by measuring AMPA and MDA ratios relative to fast spiking cells and measuring an AMPA GABA ratio, maybe they do an NMDA GABA ratio, I don't remember. They can get an idea of whether GABA tone is changing or not. You gotta move over, fool. Jesus. So now that they've shown you that basically the inhibitory components are not changing, which was one of the sort of previous hypotheses about how the critical period might close, was that you might just have an increase in inhibitory tone that causes a change in inhibitory ratio to excitatory ratio, and that change in a balance of inhibition and excitation might close the developmental window. But that's definitely not the case, because in the PSD95 knockout mice, the inhibitory tone is the exact same as the inhibitory tone in the wild type. So then you can't come to that conclusion. So in the next two figures, they're going to use a very cool technique, small, short, whatever it is, short hairpin RNA technique, whereby using a virus to express a short mRNA that interferes with the regular transcription of PSD95, they can essentially downregulate PSD95 expression in a wild type animal in specific cells that take up the virus. And in this viral prep, they use a GFP virus as a control. And so they're either knocking down PSD95 in a control animal or control cells, or they're looking at wild type. Now, when they do this, first figure, they look at it just from the perspective of, can we do it? And do we see more silent synapses on those neurons in slices? And they do. More silent synapses, less mature synapses, by knocking down PSD95. So if it works in slices, then the obvious thing in figure 6 would be to try and see if they do this virus treatment to a wild type animal at an adult age, can they reopen ocular dominance plasticity? And so that's what they try to do, and they are able to reopen. It's just a turn signal, that's all you need. Turn signals are great because they tell other people where you're going before you go there. But people in Pennsylvania don't use turn signals, they just don't. It's above them. I think it might even be that Trump voters don't use turn signals because they don't care about anybody else. Please go. Please go! So in figure 7, interestingly enough, after figure 6, which I think is really the only thing they needed to show, I guess the reviewers thought that there was one more prediction that they could test, which is that if what they say is true, then because monocular dominance or monocular deprivation leads to a change in wild type that then goes back when you reopen the eye, the question was, or the question that the reviewer or somebody asked was, wouldn't it make sense then that the PSD95 knockout mouse would likely remain more labile, and therefore any effects of monocular dominance or monocular deprivation would reverse quicker in that model. And that's what they show in figure 7. So it's a nice paper, and it's important from the perspective of understanding this model of plasticity, because ocular dominance plasticity has been used for years now as a very accepted model of how maturation occurs in primary sensory cortices and how we can mess with it. And so understanding how the critical period changes through the maturation of silent synapses, it was an important hypothesis for a long time, and maybe this paper makes it officially there. I don't know. But it's a nice paper. I hope you enjoy it. I hope the discussion is fruitful. Hey, thanks for joining me on my ride. That was a very interesting paper about the maturation of V1. Understanding how somatosensory cortices mature is a very important part of neuroscience, because understanding the difference between sort of maturation as opposed to growth during a mature adult's life, those are two very different learning processes. So understanding how the visual system is wired up is quite a bit different than understanding the process by which, for example, we learn to remember things or we recognize objects. This kind of thing is much higher level V1 function. What we are now talking about actually is just the maturation of the binocular version of V1. And the idea is that if you deprive one of the eyes during the critical period, instead of wiring to the contralateral eye, the visual cortex will start to accept or re-accept the signals from the open eye instead of the deprived eye. The idea being that the normal wiring is that the eyes wire contralaterally to V1, and if you then deprive the contralateral input to that side of V1, then that side of V1 will then compensate by listening to the ipsilateral eye. This is called monocular deprivation, and it's called ocular dominance plasticity. The cool thing is that this can only occur in a very short window of time during the maturation of the cortex, and so if you monocularly deprive an animal outside of this critical period, then you don't change the bias of V1's binocular region. Interestingly, the authors of this paper made the observation that a particular knockout mouse, PSD95 knockout mouse, does not show this particular ocular dominance plasticity window closing. The critical period stays open, and through a series of manipulations and experiments, they've been able to show pretty convincingly that it's the presence of PSD95 and it's the stabilization of the synapse that marks the critical period and actually functionally defines it. And if you get rid of PSD95, then you can even take a wild type animal and set it back out of its critical, or back in to its, reopen this developmental window and essentially extend the critical period or reopen it. If you liked it, like and subscribe. Thanks for watching, and I'll see you next week.